So this is just an overview of the type 1 diabetes. It is an unknown autoimmune disease. Still, they don't know what still causes it. It affects children, adolescents. It affects genetic component, which is part of the chromosome 6. Actually, the genetic component is chromosome 6. It affects on the short arm, which the short arm has a allele MHC class 2 gene, and it affects, in specificity, it affects the DR3 and DR4 haplotypes. And it also affects the pancreas, which has the beta cells within the islet. So that's what we'll be talking about. So what has been done for experiments? So I'm going to be talking about adenoviral vectors, lentiviral vectors, and I found a human clinical trial. It's mainly with transplantation. I couldn't really find any human trials that they actually successfully inserted adenoviral vectors or lentiviral vectors. But that's about it. So in this study, it focused on the adenoviral vector, and what it is, it has an icosahedral protein capsid that creates a strong immune response for many people, like including diabetics. And for this study, the researchers were trying to find out or figure out how the non-obese diabetic mice, how they, respond, and they evaluated the protective effect of the ADMIL10, and that stands for the adenoviral vector mediated interleukin 10. Using the interleukin 10, which is a cytokine, it has a presentation of function by inhibiting MHC2 molecules. And it has a role in suppressing the immune system with anti-apoptosis to stop the release of inflammation factors from the macrophages. And when doctors see a low number of interleukin 10, it's an issue in T1 diabetic patients. I abbreviated that, if that's okay. So over here in the table 1 and 2, they use mice, a total number of 24 mice, mainly females. They use females. I don't know why, just mainly females, but that's what they used. And they put them into 3 groups. They put them group 1. They were injected with 0.1 milliliters of the ADMIL10, which is the adenoviral mediated interleukin 10. And then group 2 was injected with 0.1 milliliters of adenovirus vector without the interleukin. And then group 3 was the diabetic control. And then it shows in the tables, table 1 and 2, that it shows that group 4 is involved too, but they only use it as a normal control, meaning they had no signs of being diabetic, and they used 8 mice, but injected them with phosphate buffered saline, which doesn't really give any effect. They're normal to see how they function normally. So these mice became diabetic within 3 days, and when they did that, they used them. They use them after the 3 days. And so as you can see in the tables, group 1, they had their reduced body weight. So table 1 shows their body weight on the top and then blood glucose levels on the bottom. So body weight, group 1, their body weight was lowered. Group 1, group 2, and group 3, their blood glucose levels, as you can see, were similar. In group 3, they drank more water and urinated more. They were tired, restless at week 3. If you can tell, there was a difference in week 3. But overall, within the weeks 1, 2, and 3, they were saying that all their blood glucose levels were similar. And in table 2, they showed that all blood glucose levels were similar, and there was no significant difference. In the figure 4, it shows they wanted to show the effect of the adenoviral vector mediated interleukin 10 on pancreatic islet cell apoptosis. So as you can see on the y-axis, the rate of the apoptosis of the beta cell, and then the y or the x-axis, you see the different groups that are involved. The normal control, you can tell there's a decreased number in the beta cells compared to groups 1, 2, and 3. But there was still no significant difference of the apoptosis rate within groups 2 and 3. So in conclusion, the results they really focused were just the body weight and more importantly, in the blood glucose level of the rats. And there was minor similarities and minor differences. So in this slide, it shows the composition of lentivirus. A lentiviral vector is a retrovirus, and as in the article that I found, it stated that it transduces non-dividing cells as well as dividing cells. It's like a sort of attraction characteristic that creates transduction of many cell lineages, so in doing so, that it has an expression of insulin. So I show you this figure because there's two differences in the two compositions of the lentivirus. So in the one to your left, it's called PLV CMV insulin, and then the one to your right is called PLV CMV GFP. So the difference between the two is it has different segments, as you can see, different colors around the circle, and it shows that between the CMV and the PRE, it shows that there's something different between the two, which is one has the insulin aspect and the other has a GFP. What a GFP is that it's a green fluorescent protein, and it's considered a protein marker, which shows like a color of green, and like shows the fluorescent color. These different sections are actually really important, and just that difference within the two, the one to your left and the one to your right, just the difference between those two can that's how they create it, if you will. So what I'm going to show you within the studies I found, that there's different studies that include mice, and that they show either or. They show the one to your left or the one to your right, or both together. So I'll be showing you that soon. So in this study that I have found, stem cell and gene therapy were incorporated together, and they wanted to see it can be a new route for insulin production within the beta cells. This can alleviate the autoimmune reaction within it as well. And so they wanted to see like what would happen. So what was used was the human adipocyte-derived stem cells that were transducted, which means a process by which foreign DNA is introduced into a eukaryotic cell by a virus or viral vector, which in this case would be a lentiviral vector that had a furin cleavable insulin gene, kind of like similar to what I showed you previous, that segment that I was telling you about. And then this study wanted to see the measurements or levels of insulin before and after the adipocyte-derived stem cells, and to find the presence of or the absence of the promoter AP2, which is the adipocyte-specific promoter. So to the side, as you can see, I did abbreviations. MC stands for microcarrier, which involves the mice, the diabetic mice. And then AD standing for the adipocyte-derived stem cell. AP2 is the adipocyte-specific promoter. And then INS meaning insulin. So they added that all together. And EGFP cells, which is which stands for the enhanced green fluorescent protein. So I did give a figure right here. As you can see, figure 6 shows that the researchers injected the diabetic mice in the upper inguinal fat area. Some mice were treated with the microcarriers of cytopore 1, which they had hyperglycemia. The other mice that were treated were with AD, AP2 insulin, and the EGFP cells. So, and they showed significant decrease in blood glucose levels, 14 to 28 days after treatment. As you can see in the graphs A, B, C, they show the significant differences within the if you want to say four groups, but I would really focus on the three groups: the MC, the cell, and the cell and MC, which they added them together. But the important graphs here that they really focus on were the E and F graphs, because the red and the green for graph E, day 14, they take the MC and cell, and the MC and cell with the they just took them out. They just like took it out of the mice. I don't know how. They didn't really like say, but they just like took it out. And when they did that on day 14, and they were monitored during day 17, they were monitored. But the results that they indicated transplanted functional cells on microcarriers could contribute to long-term increased diabetic hyperglycemia. So there's two different figures, and they're each from two different studies. And I put them on this slide to show you that these are also other studies that they used mice with the lentivirus, the one with inserting insulin, or the EGFP, or both. But figure 1 does show that they use the insulin lentivirus, which had a stable blood glucose levels, as you can tell from days 5 to 30, or either throughout the year: 3 months, 6 months, 9 months, 12 months, as you can tell in the figure 1A and figure 1B. That's what they were really striving for, to see if it was the same. And I incorporated these two together because they both showed two different ways they did it, but it was with mice as well. Figure 2, it actually shows 4 different groups that they used, and it was similar to the previous slide, that they used different groups, but they just wanted to see the fasting blood glucose levels, and to see if the administration of the EGFP was like of any interest. And so with both of these, what they resulted, and with the other study, that administering EGFP lentivirus, it does not cure diabetic mice. So in this study, it's actually interesting because it uses pigs instead of mice, which I found very interesting because like they're using the same aspects of how they experimented on the mice. It was just different to how they did it. There were 3 important things that they wanted to involve in a stepwise fashion, which, for example, included normal blood glucose levels for a period of time. The second one was, once normal blood glucose levels were achieved, the next process was having a certain amount of lentiviral vector that had the INS FUR, which means, or would stand for furin cleavable human insulin. We talked about that, which was part of the lentiviral segment. And then intravenous glucose tolerance test that had to be achieved within 30 days. But the third main thing that they wanted to achieve for this whole study to be like legit, I guess, if you will, or more along the lines of like able to study or experiment, which was the third thing, was after maintaining the blood glucose levels and the insulin furin cleavable human insulin, then there was an observation of the reverse transcription polymerase chain reaction, which analysis of the pig's liver was expressed either an adequate amount of beta cell transcription factors, pancreatic hormones, and glucagon. So they only used 9 pigs. If you really when I seen this study, like 9 pigs, and out of the 9, they really focused on just one. And I put pig 723, which was the number of the pig that number one pig that they really focused on. They used this pig in question because they not wanted, but they used this one pig, pig 723, because it was the only pig that had shown normal fasting blood glucose levels. And they wanted to approach this really fast. And when they did that, they used the graphs, as you can see in figure 1. They used what they experimented on the pig. They used him or her, whoever it was. But in figure 1A, it showed that blood glucose levels of the pig that had normal fasting blood glucose levels for 10 to 44 days. The insulin given was on day 8, as you can see on the graph. 1A, day 8, there was a decrease in blood glucose, and they gave the pig 3 units. And on day 9, there was 2 more units given. And for successful results given of normal blood levels reached, as you can see, and they say till day 44. So this study really just mainly lasted only for 44 days. And I will tell you why later. But in figure 1B, it shows that with the injection of the lentiviral vector, there was a normal intravenous glucose tolerance test, the IVGTT, at 30 days. As you can see on the graph, the x-axis, the time, and then the y-axis is the blood glucose. It shows 2 different lines with the squares or the diamonds showing the normal, and then normal being this just a square, and then the HMD, insulin furin cleavable human, furin cleavable human insulin. I don't know why they did that twice, but that's how it is. But they showed on this graph, it shows that there's a decrease in the blood glucose level within the amount of time that they had, within the amount of time that the pig 723 had the injection of the lentiviral vector. And in conclusion, why I say that this test only lasted up to 44 days is because after 44 days, the pig died. And what I read in the article or the journal of their study, what I read was that it only lasted 44 days, but because the pig died. And the pig died not because of the lentiviral vector, supposedly, but they said that he died of some other cause, which I don't know if I believe that. And then the results also suggested that increasing or lowering the dosage of the vector did not result of diabetes reversal in the pigs, and that they also concluded that further testing still needs to be done, which is probably with any of these studies. So I added this human study, which was just mainly the islet transplantation that they tried to see if it worked or not. And I inputted a table showing the patients how they were grouped. Table 2, for example, table 5 to the left is actually showing the what type of patients they used and what their history of previous transplantations were. And so it shows you an overlay of what patients they used. And then in figure 3, it just shows the outcome of the HbA1c levels that most doctors look at. So I just incorporated that to see a different outlook. And these are my sources.